Hi guys, welcome back to the IB Biology revision channel. Today I'm going to be doing 1.3, which is membrane structure. So to start with, our essential idea is the structure of biological membranes makes them fluid and dynamic. So a phospholipid bilayer. A phospholipid is a lipid molecule with two fatty acid chains and a phosphate-containing group. A bilayer is controlling the passage of substances across surface exchanges. So, in a phospholipid bilayer, the bilayer is held together by weak interactions of hydrophobic interactions between the tails, which allows membrane fluidity and flexibility. Within the phospholipid bilayer, there is a polar head, which is wet and hydrophilic, and there is a non-polar tail, which is dry and hydrophobic. Together, these two elements make an amphipathic molecule, which simply just means regions of hydrophobic and hydrophilic areas. And hydrophilic means water-loving, and hydrophobic means water-hating. And here is a diagram just to visualise the phospholipid molecule. And phospholipids are embedded with proteins. These can either be permanently or temporarily attached. So the integral proteins are permanently attached. They are typically transmembrane, and the peripheral proteins are temporarily attached by non-covalent interactions, and they associate with one surface of the membrane. So now we're going to move on to the functions of membrane proteins. This can be memorised by the acronym of JETRAT, which you probably already know. Okay, and JETRAT stands for junctions, enzymes, transport, recognition, anchorage, and transduction. So I'm just going to quickly explain which each of these means. So junctions is connecting the two cells together. Enzymes is pretty straightforward, it's just the chemical reactions. Transport is facilitated diffusion and active transport as examples there. Anchorage is the attachment point for the cytoskeleton. Transduction is a function as receptors for peptide hormones. Okay, so now we're going to move on to cholesterol. And cholesterol is an amphipathic molecule, which we previously established is regions of hydrophilic and hydrophobic. So cholesterol is found in animal cell membranes, and it functions to improve stability and reduce fluidity. That's quite a nice way to remember it, as it kind of rhymes, so stability, fluidity. And here are some other beneficial things which cholesterol functions. So it immobilises the outer surface of the membrane by reducing the fluidity. It makes the membrane less permeable. It prevents crystallization of the membrane, and it helps to secure the peripheral proteins. So now we're moving on to the fluid mosaic model, which is a model of the phospholipid bilayer. The fluid component is that phospholipid bilayer is viscous, which means it's not runny, it's thick, and individual phospholipids can move position. The mosaic part of this is the phospholipid bilayer is embedded with proteins, resulting in a mosaic of components. Here are the three main points of the fluid mosaic model, which are the phospholipids, the cholesterol, and the proteins. The proteins can be subdivided into three categories of channel proteins, carrier proteins, and glycoproteins, and these are the components of the plasma membrane. However, the fluid mosaic model was not the only one which was previously established. The Davson-Danieli model, which postulated that two layers of proteins flank the phospholipid bilayer, they suggested and assumed that all membranes were of uniform thickness, all membranes would have a symmetrical internal and external surface. They didn't account for permeability of certain substances, and the temperatures of the membrane which solidified did not correlate with the expected of this proposed model. So I've just summarised it into four main points, which is the thickness, symmetry, permeability, and temperature, which this model did not actually represent correctly, and therefore the fluid mosaic model was favoured and this model negated. So the falsification of this model is biochemical evidence, which says that not all membranes have a constant ratio. Fluorescent antibody tagging, which membrane proteins are not fixed in one place. And freeze fracturing reveals rough and irregular internal surface, such as transmembrane proteins. Oh, didn't even click that one. And this can kind of be remembered as BFF if you want to do it like that. So those are three ways of falsification evidence. So that was just a quick summary on topic 1.3, which is membrane structure, and in this video we've established phospholipid bilayers, functions of membrane proteins, cholesterol and components of the plasma membrane, and the different models. I hope this video has helped, and make sure to smash that like button, subscribe and drop a comment. See you in the next video.